In the vast universe of pharmaceuticals, where precision and safety are paramount, there exists a concept that holds immense significance: log reduction. This seemingly simple term carries the weight of ensuring the sterility of medications and, ultimately, safeguarding human lives. Imagine a world where medications, instead of healing, could potentially introduce harmful contaminants into our bodies. A terrifying thought, isn't it? That's where log reduction steps in as a guardian, a measure of our success in eliminating these unseen threats. Log reduction, in essence, quantifies the effectiveness of a sterilization process in reducing the number of microorganisms present. It's a numerical representation of our fight against contamination, a fight we wage relentlessly to ensure that every dose of medicine administered is a step towards better health, not a leap into the unknown. Just like the vastness of space, the world of microorganisms is teeming with unseen life. Log reduction is our compass, guiding us towards the ultimate goal: a sterile, safe, and healthy future. To truly grasp the concept of log reduction, we need to delve briefly into the world of logarithms. But fear not, for we're not venturing into the depths of complex mathematical equations. Instead, think of the logarithmic scale as a powerful magnifying glass, allowing us to visualize large changes in numbers in a more manageable way. Imagine you have 1,000,000 bacteria. That's 1 followed by 6 zeros. Now, picture trying to track the reduction of this massive population as a sterilization process takes effect. It would be like trying to count the stars in the night sky, a daunting task, to say the least. Here's where the logarithmic scale comes to our rescue. Instead of dealing with these astronomical numbers directly, we use logarithms to represent them in a more convenient way. A log reduction, denoted by the unit log, represents a tenfold decrease in the number of microorganisms. So, a 1 log reduction means we've eliminated 90% of the initial population, leaving us with 100,000 bacteria. A 2 log reduction knocks off another 90%, leaving 10,000, and so on. It's like shrinking the vastness of space down to the size of a star chart, making it easier to navigate and understand. Now that we have a grasp of the logarithmic scale, let's see how different levels of log reduction translate into real-world scenarios. 1 log reduction. Imagine a bottle of juice containing 1,000 bacteria. A 1 log reduction would eliminate 900 of them, leaving 100 behind. 2 log reduction. A 2 log reduction would reduce the bacteria count from 100 to 10. 3 log reduction. A 3 log reduction would bring 10,000 bacteria down to 10. 4 log reduction. Visualize 1,000,000 bacteria on a surface. A 4 log reduction would leave us with 100. 5 log reduction. A 5 log reduction would decimate their numbers, leaving 100 survivors. 6 log reduction. Starting with 1,000,000,000 bacteria, a 6 log reduction would theoretically leave us with a single survivor. As we move from 1 log to 6 log reduction, the impact is exponential. In the realm of pharmaceuticals, where even a single microorganism can have dire consequences, 6 log reduction stands as the benchmark for sterility assurance. It represents a level of confidence that gives both healthcare providers and patients peace of mind. Think of it this way. If we were to administer a medication that had undergone a 5 log reduction, there would still be a theoretical chance of 1 contaminated dose out of every 1,000,000 produced. While this might seem like a small risk, in the world of pharmaceuticals, even 1 is 1 too many. 6 log reduction, on the other hand, pushes the boundaries of sterility, aiming to eliminate all viable microorganisms. It's like launching a spacecraft into the vast expanse of space. We don't just want it to reach orbit, we want to ensure it has the fuel and trajectory to reach its destination safely. Section 5. A real-world example. Ensuring the safety of injectable medications. Perhaps the most relatable example of 6 log reduction's importance lies in the production of injectable medications. Unlike tablets or capsules, which pass through the digestive system where some level of microbial defense exists. Injectables bypass these natural barriers, delivering their contents directly into the bloodstream. Imagine a scenario where a batch of injectable medication is contaminated with bacteria. If this product, having undergone only a 4-log reduction, were to be administered to patients, the consequences could be devastating. Now, picture that same scenario with a 6-log reduction process in place. The likelihood of even a single bacterium surviving the sterilization process is infinitesimally small, virtually eliminating the risk of contamination. It's like the difference between sending a message by carrier pigeon and transmitting it through fiber optic cables. The latter offers a level of speed, reliability, and accuracy that the former simply cannot match. Section 6. Beyond 6-log reduction. The quest for absolute sterility. While 6-log reduction represents the gold standard in pharmaceutical sterilization, it's important to remember that it doesn't equate to absolute sterility. The complete elimination of all microorganisms, including their dormant forms like spores, is a near impossible task. Think of it like trying to catch every single grain of sand on a beach. You might be able to collect the vast majority, but there will always be some that elude your grasp. However, the pursuit of absolute sterility doesn't end with 6-log reduction. Scientists and engineers are constantly researching and developing new technologies and methodologies to further minimize the risk of contamination. It's a continuous journey of improvement, driven by the understanding that even when dealing with the invisible, the stakes are incredibly high. Just as astronomers continue to explore the universe, seeking answers to fundamental questions, so too do pharmaceutical professionals strive to push the boundaries of sterility, ensuring the safety and well-being of patients worldwide. Section 7. The role of validation in achieving 6-log reduction. Achieving 6-log reduction isn't simply a matter of subjecting products to a sterilization process and hoping for the best. It requires rigorous validation, a systematic and documented approach to demonstrating that the chosen method consistently delivers the desired level of microbial reduction. Pharmaceutical manufacturers must validate their sterilization processes using scientific principles, engineering controls, and statistical analysis. Selecting the appropriate sterilization method, different methods, such as heat, radiation, or filtration, are effective against different types of microorganisms. Monitoring and documenting the process. Each sterilization cycle is meticulously documented to track performance and identify any deviations. Validation is the backbone of 6-log reduction, ensuring the sterilization process is robust, reliable, and effective. Section 8. Regulatory requirements and guidelines. Given the critical importance of sterility in pharmaceuticals, it's no surprise that regulatory agencies around the world have established stringent guidelines and requirements for manufacturers. These guidelines, often based on scientific consensus and industry best practices, provide a framework for ensuring that medications meet the highest standards of safety and efficacy. One such prominent regulatory body is the U.S. Food and Drug Administration, FDA. The FDA mandates that sterile drug products must be manufactured using a validated aseptic processing system. This system must be designed to prevent microbial contamination and must be rigorously tested to demonstrate its effectiveness. In addition to the FDA, other international organizations, such as the European Medicines Agency, EMA, and the World Health Organization, WHO, have also established guidelines for the production of sterile pharmaceuticals. These guidelines emphasize the importance of risk management, quality control, and continuous improvement in ensuring patient safety. Compliance with these regulations is not optional. It's a fundamental requirement for any pharmaceutical company involved in the production of sterile medications. These regulations serve as a safety net, protecting patients from the potentially devastating consequences of contaminated products. Section 9. Conclusion. 6-log reduction, a cornerstone of patient safety. In the intricate and ever-evolving world of pharmaceuticals, 6-log reduction stands as a testament to our commitment to patient safety. It's a testament to the countless hours of research, development, and validation that go into ensuring that each dose of medication administered is free from harmful contaminants. As we continue to push the boundaries of medical science, developing new and innovative treatments for a wide range of ailments, the importance of sterility assurance will only continue to grow. Just as the stars in the night sky guide lost travelers, 6-log reduction serves as a beacon, illuminating the path towards a future where medications heal, not harm. It's a concept that underscores the interconnectedness of science, technology, and human health. A reminder that behind every successful treatment, behind every life saved, lies a foundation of meticulous planning, rigorous execution, and an unwavering dedication to quality.